introductory lecture to the strategies to engineer extracellular vesicles and provide a kind of a background to the total force speed that follow. As I said, my name is Amir. I'm a professor at Karolinska Institutet in Stockholm. My lab is on extracellular engineering or EV engineering for therapeutic purposes. And why are we interested in EV? You've seen these type of pictures many times these 3 days. So why we are interested in using them and why they are interesting as drug delivery vehicles is that they appear to be safe to repeat those. They have the ability, as they are lipid bilayers, to protect the RNA that is inside or proteins that are inside. They've been reported to be able to transport molecules across biological barriers. Like Richard said before, but I believe the real power of these vessels is actually that they are potent surfacing nanosomes. They contain a lot of molecules on the surface that can have very intrinsic biological effects on recipient cells. And what I would rather go more into this talk is whether what is in the inside is likely to be delivered if we don't engineer these vesicles. And of course, they are amenable to engineering. So there are basically 2 broad ways to engineer EVs, and you probably heard that on the educational day. One is what we call endogenous engineering. That is when you manipulate the producer cells to secrete EVs that carries a protein and RNA of interest. Those can also be divided in 2 different tracks. So you have passive endogenous engineering. That means that you're overexpressing an RNA of interest and hoping that you're going to get enough copies into your cyclotron. Or you have active endogenous engineering, meaning that you tether this to extracellular proteins that then drag the protein of interest or the RNA of interest into vesicles. And then there is the exogenous loading approach, which we will hear much more about in 3 of the coming talks on strategies how you can load things post-isolation of vesicles. So, you can engineer vesicles for many different purposes. The focus of today will be for drug delivery because most of the talks are related to drug delivery. But of course, you can engineer them in different ways to study biology, and I'll show a few examples of that. And this is a picture basically showing that what you can do is you can display things on the surface by tethering various domains to membrane proteins that are abundant in exosome surfaces or EV surfaces. You can tether things to the inside, like fluorescent proteins, bioluminescent proteins, in order to be able to trace them in vivo and in vitro, or with protein therapeutics like gene editors. And you can also put targeting motifs, either peptides or Fc binders to decorate with antibodies. So there are a whole slur of strategies you can use, and you can use this to enhance pharmacokinetics, you can do this to enhance targeting, to enhance luminal loading, and also to enhance release of the cargo. So this is just to show that there are also methods, of course, how you can engineer EVs to study biology inside cells, everything from regular GFP and Sherry type of labeling to study where they are going. You can lectin systems to look at endosomal rupture. You can use split GFP or nanobit. We have split enzymes. If you deliver to the cytoplasm, we get a signal to look at the cytoplasmic delivery. And then there is the emergence of all these proximity ligation assays when you can do in situ bioaccumulation of proteins to look at protein binding to EV surfaces. Then there are also, of course, functional. So one thing is to study where EVs are in a cell, but ultimately, if we're talking about drug delivery, we want to deliver it to the site of action, which is either in the cytosol in most cases or in the nucleus. And these are 2 well-established reporter systems that are used quite heavily in the EV field and also in other type of nano delivery applications. One is the CRE-LOC system, where you load CRE recombinase into EVs. You then have reporter cells that have a GFP or an RFP interrupted by a stop codon, flanked by OXP sites. And once you get recombination in the nucleus, that stop codon is removed, and you get a fluorescent protein production, in this case GFP. There are very similar systems developed from Cas9, CRISPR-Cas9, but here you can use a frame shift Intel that causes a frame shift to remove the stop codon, and then you get production of functional protein. These are 2 examples of functional assays that really tells you whether EVs have delivered their carbon, the carbon that is inside, into the interior of the cell. So what are the limitations?
3, there are many more. 4, there are many more that we heard in the previous session. But what I think one of the first issues is that when you inject EVs, they are very rapidly cleared from circulation. So the half-life in most of our studies suggests that it's in between 2 and 3 minutes, depending on how they are engineered. So they're incredibly fast cleared from the blood. And they are taken out predominantly by the monocytic phagocytic system and then degraded. So that has to be overcome, and I'm sure we're going to do that. The other problem is, if we talk about androgen problem, is to get enough of the therapeutics into the vesicles to actually be able to get a therapeutic effect with these vesicles. And I'm showing how, without engineering, what type of numbers we have and how you can up these numbers for therapeutic purposes. Once you have delivered your payload, the payload also needs to be released. So if it's stuck to a release scaffold and it's not released, it's not going to have a biological effect in the recipient cell. And the fourth, and in my opinion, one of the real shortcomings is that the rate of endosomal release of luminal EV cargo is very, very limited. So I think there is a big need to enhance this. So, if we talk about pharmacokinetics and biodistribution, as I said, the half-life is about 5 minutes. EVs mainly distribute to liver, spleen, and lungs, so there is very little of an injected dose that will go extrahepatically to other organs. And this really requires surface engineering to enhance PK so that they stay longer in blood, to also give a chance of targeted delivery. And there are various ways to do this, and these are just some examples from literature. These are from my lab, where we have exposed EVs. We've engineered them to bind albumin by inserting albumin binding domains on the surface of vesicles. By doing so, we can enhance the circulation time quite dramatically. Another strategy that has been widely used is to use don't-eat-me signals, like CD47, CD55, that also enhances or avoid some of the early macrophage clearance. And then there are exogenous ways, so you can use different lipid tags, either pegylate EVs, or to, again, small molecules to bind albumin with high affinity, and this is something that we published last year, that also enhances the circulation time. So, now talking about surface modification, and in general, doing engineering on the surface of EVs is fairly simple. We've identified most of the in the field knows that tetraspans are really abundant. There are other single transmembrane receptors that are abundant on the surface of EVs that you can engineer and impart new functions. It's way more tricky when it comes to luminal loading. I would start by showing some of the work in the field on RNA loading, because most of the talks in this session is on RNA. And one of the, I like this, is a very recent review from Gerd Strok's lab, where showing that if we look at RNA in EVs, if we use methods of precipitation, for example, you find a lot of RNA. So if you were to do precipitation all the time, you're going to identify a lot of RNA. However, if you do size exclusion, where you fractionate based on size, you see that RNA amounts are very low. They are underrepresented in the secretome in vesicles. And when we did some early work on small RNA sequencing, this is work from one of my students a couple of years ago, we found that most of the surface-treated microRNAs are indeed non-vesicular, and only a very small portion, when we measure by size exclusion, are vesicular associated. And even if we overexpress a prime microRNA or an shRNA, so short for terpene RNA, we see the same. The vast majority of secreted small RNAs are non-vesicular. So that brings in a problem to the equation. How do we actually use this to functionally deliver small RNAs? Because if you do the calculations and you have an average size of an EV is 100 nanometers, this is the total percentage of a cell volume. And in that volume, you can accommodate between 70 and 20,000 RNAs in total. And this is the literature that we base these numbers on. But then the next issue is that the predominant species of RNA is not microRNA or mRNA, that are usually the therapeutic RNAs that we want to deliver. It's rather liRNA, siRNA. And actually, if you look at the copy numbers of individual microRNAs, they are maybe 1 in 100 EVs. And with mRNAs, what we see when we do absolute quantification of intact mRNAs, you're looking at about 1,000 copies in 1 million EVs. So there is a number problem. Because we now take those EVs and we can put them on recipient cells. Let's say we have 10,000 cells, we put them on to the power of 9 EVs, and we count really high. You have one copy of an shRNA, of your therapeutic RNA. You have 100,000 copies available per cell. Say that you have an average uptake of 10%, which is probably highly counted. That you have 10% endosomal escape, which is about 10 to 20 times higher than what has been reported for any other delivery modality. And that you have that one copy. Then you're going to get thousands of cytoplasmic copies. And with the best sRNAs in the field, chemically modified, the cytosolic IC50 value of an sRNA is about 2,000 copies sRNA per cell. So we have a problem. We don't even have enough copies to reach an IC50 value. And this is why you need engineering, to really up these numbers. So there are different ways to do that. You can either overexpress, again, the passive loading, an shRNA or an mRNA gets passively loaded. You do have active loading, either that there are motifs in mRNAs or microRNAs that are preferentially recognized and sorted into vesicles, or you fuse an exosomal protein to an RNA binding domain that then drags your RNA of interest into the vesicles. And that's the active loading. Yes. And I'm just giving one example. This is you can read more from Julia Radler's poster tomorrow, poster 151. And this, what we've done here is we've done a quantification of shRNAs that we've overexpressed targeting the MPH. And what you can see here is that if you only overexpress an shRNA, we don't even have one copy in 100 EVs. If we overexpress it together with our RNA binding domain, AGO2, you up that number quite significantly. And if you now put an EV sorting domain, you're nearly approaching one copy of shRNA per vessel. We then put that on recipient cells, we'd see nice dose-dependent production of gene expression. If we have a passively loaded, overexpressed EV, there is no silence. So, this brings me to the talks that are going to follow here, is that one way, of course, to enhance this, if we don't reach enough numbers of small RNAs, is to use exogenous loading. And that can be done by electroporation or sonication or simple co-incubation, but it can also be using CPP-assisted, so peptide-assisted loading, or nanofluidics mechanoporation that we will hear more about, or tonicity brain osmotic loading. These methods are much better at enhancing loading of small molecules and even short RNAs. And they are efficient, you can control the dose, it's fast, and there's no need to cargo release. There's nothing stuck to it. You're loading something exogenously. The limitation is, of course, that you need very high concentrations of your payload. And until to date, it's only been efficient for smaller payloads. So going for larger proteins is very challenging using exogenous loading. And these are the three talks that will deal with how this new methods can enhance this exogenous loading. And then when it comes to protein loading, as I said, exogenous loading is really challenging. You need to go for an endogenous approach. And to do so, what you do is that you need a membrane protein or a protein that is ferried to the vesicles, and it can be CD63, and it needs to be fused to the cargo, so the protein. That could be Cas9, for example. And you need something that can cleave the cargo from the protein. And this protein can also be a pyruvation task. And that's what we've done here also, the second lab speaker today, how he has used this system to load the CRISPR-Cas9 into vesicles. The problem is still there are two remaining obstacles that I would like to bring attention to, and I think it's really important to keep in mind. It is cargo release and endosomal escape. And what do I mean with that? This is typically how it looks like. When you do dose response experiments and you put EVs, GFP-labeled EVs, on recipient cells, and we do storm imaging of late endosomes and lysosomes, we consistently see this pattern of endosomal swelling. So the more EVs you put into the system, you get more swelling of endosomes, but we don't detect any release events. We don't see escape events of those just sitting in those structures. And that's a problem, because we're not going to deliver something to the nucleus to the cytoplasm, but they are sitting in here It's not going to work. What we have done in my lab is we've developed a system where we have a EV sorting domain, we load with Cree or Cas9 or any other protein of interest, and we introduce a pH-sensitive cell-sliced protein sequence called a median cleave. So when pH drops during biogenesis, it cleaves. If the pH doesn't drop, it still remains tethered to the Cas9 protein or Cree protein. Now, what I want to bring your attention is the importance of having this linker, but also that you need something that enhances endosomal escape. So if we now take these EVs, we take EVs loaded with Cree, the EVs that we have engineered to have CD63 Cree, so we get more Cree into the EVs. We have an INTIN only that cleavable linker to Cree, or we have CD63, the cleavable linker and Cree. So theoretically this one, we know it loads really well, we have a lot of Cree recombination of those vesicles. But if we look, we put enormous amounts of EVs on these recipient cells, HeLa cells, P47B or B16, we see absolutely no recombination. Doesn't matter what source of EVs, what source of recipient cells, the level, the frequency of recombination is enormously low. Now, if we do the same experiment, but we include the fusogenic protein, taking inspiration from viruses, because that's what envelope viruses have learned to do. They escape endosomes to replicate. Then it looks completely different. Again, if you have Cree together with this fusogenic protein, BSVG, we've tested a bunch of others, and there are hERG proteins and cytins that people are working on. But basically what you see is that if you have Cree only and the fusogen, it doesn't work because you don't have enough Cree in the vesicles. If you have CD63 Cree plus the fusogen, it also doesn't work because you don't have cleavability. If you have INTIN Cree and the fusogen, it doesn't work because you don't have enough loading. But if you now put your EV protein, the cleavable linker, and the recombinase, and the fusogenic protein, you will see that you max out recombination. So to me, that tells us that you need, if you're going to go down the route of loading of protein therapeutics, you need releasability, you need to release it, and you need to enhance endosomal release. And this work you can find in this preprint. So, these are my conclusions and like pitfalls and also challenges to tackle when it comes to EV engineering. First of all, I think we need to find alternative ways to mitigate macrophage uptake and fast plasma clearance, because if you have very fast plasma clearance, you will never get enough of your payload to the tissue. That could be using exogenous modification. Your protein loading is, I would say, solved largely in the field. You can get hundreds of copies of proteins into a vesicle, but getting enough RNA in a region is a real challenge. And that is particularly true when it comes to guide RNA and Cas9. And this is not only in EV field, this also applies to virus-like particle when they work from David Liu at MIT. They also struggle to get enough guide RNA copies in. Of course, BSVG is not ideal. You can't repeat those. You don't want to use viral fusogenic proteins. So it's going to be a big challenge would be to identify human proteins that are fusogenic, like syncytins. In our hands, so far, they have not been efficient enough. And then, of course, one should be mindful when you go for a local or systemic delivery approach. I think you always need to let the numbers guide you. EV particle doses are not good enough. It doesn't matter how many particles you have. If you don't have enough payload, you can put as many particles as you want. And we should start really performing dose responses based on payload. So if we want to deliver Cas9, we give the concentration of Cas9, not 1 E9 vesicles. And then, of course, have the accurate controls. And with that, I leave the word to Samir.
Selected to present his work, Thomas Hector from Conrad Research Center for Natural Sciences. He will talk about the thinking-based utilization of antimicrobial and self-penetrating peptides in vesicle interactions.
Thank you. First I would like to thank the organizers for giving the opportunity to present the results here. Just a quick shout out to Australians. First time in Australia, everyone is really chilled out. It's a really nice area. Maybe you're not surrounded by countries, that could be just, I don't know. But really happy to be here. Okay, so I'm coming from Hungary, from Budapest, and as you already mentioned, from the Research Center for Natural Sciences. And you can you see my pointer here? You see the pointer? Okay, I use a basic pointer instead. So, our campus is located here. We are very happy to be in the vicinity of 2 additional universities, which gives us a really vibrant environment. So, our group focuses on 3 major directions. In principle, we work with natural antimicrobial membrane-active peptides. We also work with the non-natural derivatives of these. In principle, we'd like to go mostly towards killing microbes in principle. And since these are membrane-active systems, we also need membrane systems, and that's why, to an increasing extent, we started to work with extracellular vesicles as well. Of course, these 3 directions are quite interconnected. So, the main motivation of our direction is antimicrobial resistance. So, not necessarily going to do directly the loading challenges, but you will see that some of the things I think we do is actually directly directed or related to that. So I'm not going to introduce antimicrobial resistance. I only want to point out that by now it's not only a human health threat, it is also an agricultural problem. We have pesticide resistance and insecticide resistance already due to the overuse of small molecules in agriculture as well. So we really need to move in the antimicrobial regime to alternative compounds, and antimicrobial peptides are just potential for that. They are larger than small molecules, have a bunch of benefits. They are around for about 1 million years for some of them. We really see in the last 15 years how over 70 peptides that have been approved, so they are a good drug potential, or they are potentially good drug candidates. And we also see, though limited number of studies, that their resistance against them is developing much smaller. Of course, also drawbacks, enzymatic susceptibility, and much lower activity than small molecules. So this is where potentially EVs could come in. But I want to point out that most of them target the membrane, and this is kind of connected to some sort of oligomerization. But of course, this also means that as part of the immune system, they are going to interact just intuitively with a bunch of other components that are going to be also part of the immune system. And of course, recently we have a growing number of evidence showing that EVs are also having antimicrobial functions. This is just a recent review describing both human and plant EVs, primarily as delivery agents, as we have seen in the introductory talk. So, as I said, most likely they interact also in a natural system. We have over 2,000 host-antimicrobial peptides. So their interaction, in order to get this in a systematic manner, is going to be kind of relevant for general biology, molecular biology inside as well, but also for the translational directions, as we heard in the introductory talk, towards cargo delivery or towards cargo loading, especially if we could use not EMPs, but for instance, cell-penetrating peptides. So what we have done, we really didn't understand the systematic manner. We don't really have an overview of these type of interactions that could occur. So what we have done, we've selected a representative set of AMPs, including several cell-penetrating peptides, high charge, low positively charged peptides, and we took red blood cell-derived EVs because those are really good for biophysical studies. So throughout, I'm going to just use this orange thingy for representative of AMPs, and my animals are going to look like that. Okay, so we kind of addressed and focused on 2 directions. One is surface interactions, especially in view of the recent, of course, really by now established evidence that we have a protein corona around the EVs. This is particularly really unique, and it's not the same as using, for instance, synthetic liposomes when we check membrane interactions. And also for potential vesicle damage, we focused on their EV disrupting affinity, because many of these really target negatively charged membranes, and most of the EV bilayers are going to be partially negative. So I'm going to just focus on these two areas separately. And what we have done here, we use several techniques, and one of them is going to be this unique spectroscopy technique called linear dichroism. I'm not going to go into the details, but the key takeaway is that it's able to report on components, molecules that are either absorbed on the surface of the bilayer or inserted into the bilayer, as long as they are oriented. And the good thing about red blood cell derived EVs is that we have, as part of the protein corona, here you can see the time images, we have hemoglobin on that, so they are red, and we have a really nice foray band due to the hem group at 420 nanometers. So what we have done with the initial example when we tested this with an AMP, nalipin in particular, is that we added that, and what you can see is that compared to this rod surface, which we have for the control EV, once you have the treated EV, then actually that kind of removes the protein corona from the surface. So at the same time, for the same sample, the 420 nanometer absorption peak is going to disappear. So schematically, something like this is going to happen. We have the AMPs, and they kind of, in an optimal, of course, ideal world, they just shift these away. So this is what we have exploited, because taking mass spectrum takes about 3 minutes, which means that it's a really good technique for doing titration. So what we did, we took these peptides and gradually increased the concentration, and with that we kind of followed what is going to happen on the surface of the EVs. So we did this for all 26 of the peptides, and what you see here, this is the relative intensity of the original peak, and as we add more and more, it starts to decrease. And immediately with this, we kind of had 3 different categories to identify. Some of them are going to be even at very low concentration, absolutely removing the protein corona, so they have a high protein corona removing affinity, whereas for others we had to use over 200 micromolar concentration in order to get this peak disappear. So we went on, of course, confirmed with this just by sampling with TAM, and you can see that some of them need only 5 micromolar to make the surface clean. Others need 50 micromolar, so much higher concentration. But in an in vitro term, we could kind of identify this clearance with TAM as well. So then we went to check zeta potential and measure that, and I'm not going to talk again about zeta potential background, but what you can see is that the revs, maybe you do not really see this here, the revs are typically minus 30 millivolts. That's where they are, minus 32 plus minus 0.71 for us to be precise. And so once we start to treat these with these 7 selected peptides that we just had here, you see that even at the low concentration, this drops significantly to like minus 5, sometimes even positive. Initially we thought that this is actually due to the charge. This one in particular, F37 is plus 7 charge. But then later we kind of analyzed the data, and you can see that some of these peptides are going to have only plus 1 charge, and they can also be moved though at a higher concentration. But others, like cell-penetrating peptides, they're not really going to alter. And the reason being, we think, is that most likely the change of the surface charge is due to the removal of the protein corona itself. So once we have tested the surface, we kind of already started to categorize these into different subsets, and then we went for the EV disrupting affinity of these. So what we did FCM, flow cytometry for that, and you can see we have done this for all 26 peptides, but I just select 7 for demonstration. But some of them at lower concentration, they're not going to particularly damage the vesicles. We lose about 15 to 20% of the vesicles in general. But others at the same lower regime, they're going to pop up pretty much 60% of them. Whereas if we go to the other side, for cell-penetrating peptides, we see that even at 160 micromolar, we don't really have a significant lysis compared to the others. So we have done this with MTA. I'm not going to talk about the MTA because if you just compare the two, this is schematically really similar, and this is how it's supposed to be, so we were really happy with that. And of course, again, we had some TEM images which actually confirmed that while some of them, even at 80 micromolar, like the cell-penetrating penetratin, is not going to remove the protein corona. Others are going to do this huge aggregation, and we have this really funky opening sometimes into layers of bilayers instead of a spherical one. So once we had this, we just gathered the data, and what we did, we plotted this as a 3D plot. So we have FCMD for all 26 of them, LD data for all of them. So one represents the vesicle disrupting ability, the other represents the protein corona removal ability, and we plotted them against concentration. So we got this 3D plot, and out of that, we were able to kind of categorize the different parts. So we managed to have 4 different major categories for these, which we further split it into 8 subset categories. And you can see that these ones, we kind of point flipped the term surface cleaners, very low concentration needed, immediate clearance of the protein corona, but they are not really harming the vesicles at this low concentration. Sorry? 1 minute? Oh, shoot. Okay, I'm trying to speed up. Okay. Okay, so as you can see, we have the other, these one are going to be typically cell-penetrating peptides, and we have here those which are going to really disrupt the vesicles even at low concentrations. So if I have 1 minute, I'm really going to just rush through. We came up with a proof of principle thing for the surface cleaners, and we took LF57C and 15 and one of the FK16s from here to see whether we could engineer the surface of the vesicles. So what we did, and it turned out to be going well, is that we took proteomic total protein content for the original EVs, then we kind of removed the surface proteins with the surface removing ones, and we took for that also the total protein content. So the key takeaway is that 90% of the origin protein is still retained, so they don't really disrupt, and it kind of works. And with that, we were able to identify the protein corona members. So what we did in order to be rigid, I'm done in 20 seconds. To be a bit more rigorous for this, is that we selected those as protein core members which are removed by at least 2 of the peptides. You can see that FA16 was removed out of the total 17 which we identified, 15, and the others just do a bit lower on that. So with that, I'm kind of concluding. We have identified these different categories. I'm really hoping that this is going to help for many of you to use peptides. We are also happy to collaborate on that for various directions. I would also like to say that this is Sonalia Tasvir-Lesberg, who is here, and she's going to be here throughout the talk, throughout the conference by all the end. So just approach her. She knows all the details about the different parts. I also have to point out that we have done this on reps, and with that, I would like to thank for the collaborators, Edith Uzash, Laszlo, Lilo in particular, the whole biological soft matter platform, and thank you for your attention.
Thank you, Thomas. Do we maybe have one question from the audience?
Wonderful talk. May I ask how you purify the EVs? Where the corona come from?
So, yes, this is a very good question. The whole study was in principle, proof of principle, whether we can perform, especially if you think about the last one, or you just think in general.
In your experiment.
Okay, so in general, what we do typically, we take from volunteers blood samples,
and then we use, after that, we wait for a week. So it could be that, for instance, some of the proteins that I was showing, they are going to come due to the pop-up of the vesicles, and then after that, we typically do either centrifugation or SEP, and that's what we purify. This is the question.
I see. Yeah, I totally agree on that. Thank you.
Thank you, Thomas. Again, unfortunately, we don't have more time for questions. Our next speaker is Hui Yan from Shenzhen Institute of Advanced Technology from Chinese Academy of Sciences. And the talk will be about handcraded platform with ultra-high throughput for versatile loading of small extracellular vesicles. Welcome.
Thank you very much for your kind introduction. My name is Hui Yang from Shenzhen Institute of Advanced Technology, which is affiliated to Chinese Academy of Sciences. So the topic is regarding the nanofluid technology. I was wondering who are familiar with this kind of topic, nanofluidics. So I will try my best to introduce you, not how it works. So actually we know that EVs can be used as a very good candidate for natural drug delivery system because they have quite good advantages. So when we think about how we can engineer this, normally we have these two main approaches. The first one is the endogenous loading, meaning that we normally modify the cells first, and then we use the cells as a factory to produce the EVs, and then we have the drugs already loaded in the EVs, and then we can use it as the therapeutic materials. And the second approach is the exogenous loading, meaning that we have the exosomes or EVs isolated from downstream cells, and then we normally use the membrane premagnetization method to load drugs into the EVs, and then we use them for further applications. So if we think about the exogenous loading techniques, the common EVs process journey looks like that. So we have the we have the cells collection and also the massive expansion, and later on we use fancy techniques to isolate and also purify exosomes, and then we use membrane permeabilization method to load drugs into the EVs and then formulate those EVs and eventually use them as clinical therapeutics. So as the bridge between the upstream massive production of EVs and the downstream therapeutic applications, actually, most of the questions we ask ourselves is that, is this loading, drug loading techniques can be used for the massive production of the drug loading EVs, because it's a really because it determines either the success or the failure of the technique itself to reach the CMC point for the therapeutics later on. So since our background is the microengineer or the nano devices, so we ask ourselves that can we use the microsystems or the nano devices to tackle this kind of problem? Why we need to use the microsystem or the nano devices if we look into the size of the single molecules or the microvesicles, the nanovesicles, or even the cells. So if we use the same dimension of the structures, we can normally accurately apply the physical fields to the target of interest. In this case, we can really solve the problems on the cells or even on the nanovesicles. So in this area, actually the microfluidic technology has been heard, I think, by most of you, because it has been already widely used for the past 30 years for biomedical applications. So the microfluidic technology is characterized by the manipulation of fluids in the sub-microliter range. So it shows great prospects in biomedical research and applications, for example, for single-cell analysis, for some biomedical devices, for example, like the implantable devices, and also it has been widely used as the lab on chips for clinical diagnosis. But the question is that if we have these EVs with these exosomes with the typical size of only 100 nanometer in size, how can we do that? So we must move from micro range to the nano range. That comes to nanofluidics. So when the target is in nanoscale, the nanofluidic technology Why? Because we give the range from 1 nanometer to 100 nanometer. The molecular behavior can be precisely controlled. In this case, the manipulation of their characteristics can be realized. So if we look back about the very first time the nanofluidics meets the exosomes, I think the very famous publication is was from 2016. So the IBM Zurich published this paper, and they used some very fancy fabricated devices to generate this flow behavior called deterministic lateral displacement to separate the small EVs from the big EVs. Actually, at that time, the production rate to really purify those EVs is low because the tiny size of these nanostructures. So then, inspired by these previous works, we started thinking that can we provide some exosome nanoengineering platform to reach efficient exosome processing and also the exogenous loading through the areas of nanofluidic channels. So then we have this ExoNP. So the working principle behind of this device is mechanoperlation. So in this case, when the exosomes and also the drug substances are loaded into the chips, and then when they pass the nanochannels, so they can be slightly stretched by the compression and the fluid shear force. In this case, some transient nanopores can be generated on the membrane of the exosomes, and it gives some pathways to the drug substances to be encapsulated into the exosomes. And after a few minutes, actually the membrane can be recovered by itself. And after we got this idea, the very first question we asked ourselves is that, does the mechanoperlation cause damage to EVs? So we use some multiple different analytic instruments to verify that. The answer is no. So in this case, we use either cryo electron microscopy to verify that after being treated by this kind of nanofluidic device, the exosome membrane keeps intact, and also it shows the same level of morphology compared to the untreated ones. But probably the size is a little bit bigger because the drugs are loaded into the exosomes already. And also, the exosomes, untreated exosomes and the treated exosomes shows the same expression of protein markers. And we also use NTA, nanoflow cytometry, etc. to verify that the EV recovery yield after being treated by the nanofluidic device can reach as high as, can be higher than 95%, meaning that only a minimal amount of the EVs can be lost during the process. So then, we are curious about the loading efficiency, so we use some good model with the known size and also the fluorescent to verify that. And then we use the fluorescent extra of different molecular weights to quantify the loading efficiency. And we noticed that actually the loading efficiency is working condition dependent, meaning what? In this case, actually, if we can put up some very good designs, and if we can really optimize the working flow conditions, in this case, we can optimize the loading efficiency in this case. And then after these verifications, we change to some applications. The very first drug model we tried is the chemical drugs. We use the chemotherapeutic doxorubicin hydrochloride, which is a really well-known chemotherapeutic drug. And then we verified that with these 10 over the 8 exosomes and almost 6 micrograms of the drugs can be loaded into the exosome. We measured by the HPLC mass. So in this case, if we compare with some previous studies that for some incubation and also electroporation method, we noticed that this nanofluidic technology can reach much higher loading efficiency in this case. And also we put some quite a lot of neurological studies to verify that those loaded drugs can be successfully gave to the cells and also to the cancer cells, and also induce the cell death with also the tumor Schindler's models. And after the chemical drugs, and then we started trying on some nucleic acids and also deep molecules. So the very first thing for the PCR or even more precise digital PCR to quantify it. And we noticed a super high enrichment rate of these loaded molecules. Well, compared to the non-loaded exosomes, it can be as high as 12-fold increase. And then we try to load some long RNAs, and we use this GFP mRNA with the size longer than 2,000 nucleotides, and we can verify that the long mRNAs can be successfully loaded into the exosomes and also generate the proteins, the fluorescent proteins in the cells. So, and even we want to push the limit even further to even bigger molecules. So we started trying some well-known proteins. So we use this wild type P53 protein as the selected model. And then after the experimental procedure, we quantified by the highly precision ELISA. And then the result tells us that 10^8 MSC exosomes can load around 134 pure P15 proteins into it. And even with some bigger ones, the P53 and EGFP protein, the size is bigger and the loading efficiency is still fine. So actually, in this case, we have this good assumption that later on, if we can use some highly purified exosomes with the more particle numbers, actually, for example, with this 10^11 SUVs, the loaded protein amount can reach as high as milligrams. So this is basically the same dosage that the clinical application requires. So if you are interested into this topic, I have my PhD student here, so you are welcome to the poster on tomorrow. So I will give basically an overview or a brief conclusion. So when we think about the exogenous loading, the topics for engineers is that can we provide a versatile and robust exogenous loading platform. So in this case, the mechanoproduction compared to the electroporation is a more mild approach. And in this case, actually, the mechanoproduction can be robust, straightforward, and versatile, meaning that we can use only one step processing. We have this continuous flow mode, and in this case, we can continuously load the drug substances into the exosomes. And in this case, it can be good for the later on applications. And still, after the working principle works fine, and we want to optimize the system, so we need to, if we want to reach the CMC points, and we need to really increase the sample throughput. So when people heard about nanofluidics, what's the normal conventional nanofluidics? If we have a nanochannel with the if we have the width and also the depth about 100 nanometer, so even if you work with extremely high flow velocity, the normal sample throughput is still like 1 nanometer per minute. So what we do is that we push this boundary, we push this limit to really increase the working modules, meaning the number of the nanofluidic channels on the single chip. And we have these 140 nanochannels working all together at the same time. And we have these fancy, advanced microfabrication technologies to make the chip can really tolerate high flow rate. And at some point that we can't even find a good syringe pump to really push all those liquids in it, so we need to develop some device. So in this case, actually, what we achieved is that we have this 500,000-fold increase in sample throughput compared to the conventional nanofluidics. Right now, the device is already good enough to process 500 microliters per minute. So in this case, it's already good enough for most of the preclinical studies. So if we look into the technology, here generates a question I always ask myself: is the nanofluidics a new method for year, my work related to encapsulate the component of CRISPR-Cas9 into extracellular for bacterial manipulation. I guess everybody in this room knows what is gene therapy, so I will go further. So there are three types of technique was using in several clinical trials: the gene augmentation, gene separation, and the genomic editing. And the last one has become very popular in the past 10 years after the CRISPR-Cas9 has developed 18 years ago. And what is CRISPR-Cas9 and why this is a revolutionary of gene therapy? So let's have a look. The CRISPR-Cas9 is the endometer's gene editing technology, and it consists by two parts: the Cas9 protein and single-guide RNA. After they combine together, the single-guide RNA will guide the protein to recognize the right place and generate a double-strand break. And this break will be repaired by two ways. It's no homogeneous end-joining and the homologous direction repair. And for the first one is Rounded correction. Those with insertion, inhalation, and problem shift is like a rounded. You don't know what you got until you do the genotype. And then for the second one is like more precise editing, because when you deliver those protein, you also need to deliver the template. So with the 10 years of the effort they put on the CRISPR-Cas9, this technology has become very powerful for gene therapy. But this protein is a very big protein and they come electrical charge, so it's hampered by the delivery way. So to solve this problem, there are many vectors used to deliver them. We can say there are two types: the viral vector and the non-viral vectors. And what I want to mention today is everybody knows EVs, and we can use those EVs to deliver either the mRNA, ribonucleic protein, and the plasmid. And so we are wondering that can we encapsulate this big protein inside of the EVs to do gene editing? So as Professor Liang just mentioned, we have two ways to load the cargo inside of the EVs. So as the name implied, the post-loading and the pre-isolation loading. So in my research, I used the pre-isolation loading. I modified the Cas9 plasmid, and then I transfixed into cells, and then further I got my EVs. So we know that the EVs is a bilayer lipid vesicle. We are thinking that if we do some like a post-translation modification on Cas9, can we tag those proteins on the cell membrane so further they can be released into EVs? So we went over all kinds of post-translation modification. We found that there is a study reported that they use a palmitoylate peptide to fuse with fluorescent protein GFP and TD tomato. And their results show that they can tag those fluorescent protein on the cell membrane, and further those proteins could be released into EVs as a tracker. And so we want to know what is the palmitylation, why, and how it works to make them to tag on the cell membrane. So palmitylation modification is whereby a fatty acid to link on a substrate they contain cysteine. But if they want to link on this substrate, they need a palmitylation transfer and then named EHHC. So the first stage, this fatty acid will link on this transfer enzyme by a cell ester linkage, and then this lipid will be transferred on the substrate so that they can increase the hydrophobic and then link on the plasma membrane. So we select 6 peptides from different proteins, and we name them PP1, PP2, MP1 to MP4. The P stands for palmitylation, so M stands for myristylation. So it means that the down 4 is combined myristylation and palmitylation. Because we know that in like 2 years ago, the professor mentioned that they can successfully use myristylation to modify SPK9 and going to invade to the for some peptide we selected, they can facilitate those GFP inside of the EVs. And the problem we know that the GFP is a very small protein. They can be easier to facilitate into EV. So what we did, we continue to use those peptides to fuse the N-terminal of SPK9. It's like this way. And then after we fuse with that one, we need to verify those peptides do not negatively affect the function of SPK9. So we use a reporter cell. We translate those genes into inhaler cells. So inside of this gene, they contain a 3-poly-A. This will stop the translation of TG to metal. And if we can use the 2 single-guide RNA to successfully remove the poly-A, then the TG tomato will be restored expression. Then my cells will turn into red. So as I said just now, if I can successfully modify them, my cell will become red. So I follow this way. I follow this way, and we can see in the group we modified and also unmodified Cas9. We can detect some red cells under the fluorescence microscopy, and then we collect those cells to send for flow cytometry. We can see compared to the unmodified Cas9, we do not see any significant change in the modified Cas9. So this result tells us those modifications done to change the function, genomic editing function of SPK9. So after this one, we want to know this peptide facilitated the protein going entire or no. So first one, we extract the protein from the EVs, and then we can see compared to the unmodified Cas9, we detect more SPK9 inside of the modified group. And then we also extract the mRNA and to measure the copies of single-guide RNA. We can see compared to the unmodified Cas9 group, we detect more in the modified one. So this result told us our peptide could facilitate the component of CRISPR-Cas9 inside of the EVs. But those things can be uptaken by the recipient cells or no, we need to verify. So we still use the AI9 cells, the AI9 reporter cell I mentioned just now. And we can see from compared to like the unmodified Cas9, there are more red cells we can take in the modified group. And for the populated one, like the PP1, they reach like 10% of the gene editing in the recipient cells. But I want to mention you again, this one we need 2 single-guide RNA. It means it's a double cut, which increase the difficulty of modifying this gene. So we are thinking that can we use just a cell line only contain a single cut? Because we may have enough Cas9 or component inside the EV, but due to that cell line is not easy to modify, so we change into single cat cells. And these cells, they are constantly express the red fluorescence protein under the promoter CMV. And there is stop codon before GFP. If we can use CRISPR-Cas9 to remove this stop codon, and my cells will become red and green at the time. So I use those EVs, like I mentioned just now, transfect and collect them and treat these cells. If I can modify them, they will become green. So I incubate those EVs with the surrogate cells with different hours, 48 hours and 72 hours. And we collect the cells, send for flow cytometry. We can see with the longer incubation, like the efficiency is higher, and I reach like 40 something, I forgot, but it's for something. And after that, I also want to know, because everybody mentioned that, okay, the EV repeated is not toxic or something to our cells. So I tried 2 treatments. Like I treated the cells, after 48 hours, I passed those cells. And then I retreated the same amount of EV again. And then after that, I collected those cells in the flow cytometry, and we can see for 2 treatments, I reached like 98% of the cells were modified by the EVs. So we can, so far, I think we successfully encapsulated the component of SPK9 inside of the EV by using conjugation peptide. So I will make a short conclusion. So if you do a gene therapy, want to use EV to deliver deliver them very industry way. And for me, I modified the pathway, and then I translated them into cells, and I use the differential simplification to plate the cells, plate the EV, and treat on the double cut recipient cell and the single cut recipient cell. In double cut, I got 10.5%, and in the single cut with one treatment, I got 30 something, and for two treatment, I got 98%. So that's for all my results. And I'd like to thank my professor, Dr. Dongli, and also my colleagues. And if you have any question, you can contact me by my email, and I'm open for any question now. Thank you.
Anyone have any question? Otherwise, I have one. Okay.
Interesting, Doug. Have you tried editing an endogenous locus? You showed editing entry or GFP reporters. Have you tried editing an endogenous locus, any endogenous gene?
So far, no.
Okay.
For this one, yeah. But if we want to, actually now we are trying to use some peptide to modify tran editor. By that way, we will use the one you mentioned, but we don't know what we could get in coming days.
Okay, and the follow-up question is that what's the release mechanism of Cas9 from distillation signal? The palmitation. You showed that you have tagged with the palmitation signal.
Yeah.
And receiving the, on entering the recipient cell, how is the Cas9 released from the signal? How is it entering to the nucleus?
Maybe I think it's that word you said. Yeah. You mean after I click those EVs to train on the recipient cell, how they release?
How they release from so now it's in the palmitation signal. It reaches the recipient cell. So the protein may be sticked in the cytoplasm. They may not enter into the
Actually, for
nucleus.
the palmitation, actually they are like recycle modification. They can be palmitorated in the cells. Also, they have some depalmitorated, and after they have on membrane those cells. This is why in my group cell result you saw some wells. Actually, they contain more copies of single RNA, but their result is bad. Is worse than the PP1. I mean, maybe we were thinking that it's due to those released way. It's like different peptides, they generate different, like the efficiency of releasing, so we got different results.
Thank you.
Thank you.
I have one quick question, and I noticed, so in all your groups you have ESUD. So do you see any editing if you remove the fusogenic protein? I mean, it's more of a point. I think you're not going to see. So that's.
Yeah. But I also saw one reporter mentioned another one, maybe we can try.
Yeah, there are a bunch of
Yeah,
different
bunch of
versions,
that.
but I think it's kind of for the audience also important to see that because you didn't mention it, but it's like you are engineering also with a fusogenic protein and not only with the palmitorated Cas9. Okay.
Thank you. I would like to invite our last speaker of this session, Yong-Kyung Cho from Institute of Basic Science from Korea, and the talk will be about tonicity-driven osmotic overloading for engineering exosolar vesicles. Welcome.
Thank you for the nice introduction. My name is Yong-Kyung Cho, and I'm from South Korea. Today, first of all, thank you for staying this late. So, I'd like to introduce a new method to load sonextra on exogenous materials to extracellular vesicles. This is my disclosure, and in fact, the lab spinner having a booth on booth number 28 on this conference. So if you're interested, you may visit. So EVs, I guess I don't need any introduction, but EV carries very important materials for both diagnostics and therapies. But sometimes we want to also engineer them. But in our group, we have developed the tools to isolate EVs first. So here's the ExoBeast and ExoPrism. We utilize both size-based and exosubstrate-based separation method we developed.
we try to use these EVs as a point of care diagnostic. So we developed some nanomaterials, like in this case gold electrode, which has nanostructures and nanopolar structures, so that we can use this electrochemical sensor using just the plasma itself. Or sometimes we make EV fusion to make some new functions, like in this case we use the EV engineer to generate ATP, and so we can use it in the spheroid or 3D structures. Anyway, in this session, we were talking about many different tools to engineer the EVs, and all of the previous speakers are talking about either preloading or post-loading. When we see these questions from an engineer's point of view, yes, the needs is clear, but the method is not yet satisfactory. Both methods are pretty much provide a low yield. And also, when we did some literature survey, people report that these methods are not gentle enough. After the conventional process, such as freeze and thaw, sonification or extrusion, the outcome of the EVs shows not the same as original EVs, and their surface proteins may have damage. So big questions, is there maybe gentler method, but still loading some materials? I'm from the engineer, like a Laminar disc, Laminar chip community, and we have used this spinning disc for many different applications. And for the EV, yes, we developed ExoDisc, which has a sort of tangential flow filtration in a centrifugal platform. So if you see the movie here, the urine is loaded, and they are filtered through these two membranes. And the washing is very efficient. We can remove the protein contaminants, but not the extracellular vesicles on top of the filter. Using this concept, when we tested their yield and purity, compared to the ultracentrifugation method, our CT value was 6 times lower, which means 100 times more particles are emitted. More importantly, if we wash it multiple times, we don't lose particles, but we remove those contaminated proteins efficiently. We can tune the pore size of the ExoDisc so that we can isolate the EVs from the blood plasma with a nice quantitative measure we can do. And it's extremely sensitive, and therefore when we inoculate tumor cells in this live mouse without sacrificing them, with a small tiny amount of blood, we can see the tumor growth. That's so cool. So using this ExoDisc, we try to see whether we can use it one more time to modify the EVs. So here, if you see the movie here, this is ExoDisc. It's supposed to be moving. Can anyone push the button for this movie one? It's moving. So this is spinning, and you can see the liquid going in and out. So efficient washing. And here, after isolation of EVs, we know the loading covers in the hypotonic solution. And since the EVs are certainly situated in a hypotonic solution, their membranes are really swell. And during this osmotic shock, the materials go inside the EVs. And after that, we can quickly change to the isotonic bubble to minimize the damage. After that, you can see that we call HEV is the EV in a hypotonic solution, and high EV is when they are come back to the isotonic condition. As you can imagine, the size got increased when they are in a hypotonic solution, and their membrane permeability actually increased. Using this calcium, AM, and for bacteria, I say, if they are goes inside the membrane, the bodies is pointed, and you can see it in the HEV. But when they come back to the isotonic solution, it's the same as before. So the membrane is healed back to the original state. When we change the osmolality of the hypotonic solution, as you decrease the loading amount is increased, but not any further below to 300 microosmolality. And when you change when we load the dose and the plasma molecular extra, the optimum osmolality was even about similar. Can we repeat this process with questions? And we repeat this change it to the hypotonic, to the isotonic, ELISA surface protein didn't change much. Their cargo of RNAs and nothing changed much. Basically, they remained similar after this kinesis change process, which means probably pretty much centered. We decided to load different kinds of materials, like here DEXA with different molecular weight. Control means we do repeat the same process like the TC, tonicity control method. However, we don't change the tonicity of the liquid. So compared to the control case, yes, we can load more materials. And for this range of the molecular weight DEXA, single-strand DNA case was interesting. This control case, we couldn't see any trace. This incubation time is pretty short, like just 5 minutes. Maybe that's why, but we couldn't see any loading. But when we used this tonicity method, we could load different kinds of single-strand DNA molecules. Now we compare with different conventional methods. So here, the I means the input template, tonicity means after this tonicity control, S is for the solidification, and E is for the exclusion. When we measure number of particles, yes, it decreased significantly after this process. When we measure the surface proteins like CD9, etc., it decreases a lot, which means because we compared the same input sample volume, and therefore maybe due to the particle loss as well as the surface damage altogether in this case. So we question whether it's really coming from the yield, like loss of the particles, or it's also coming from the surface damage. So after this process, we normalize and count the number of particles and input the same number of particles as well. In that case, also we see the reduced surface proteins, which means, yes, indeed, the surface is damaged after this conventional solidification or exclusion method. Yes, so anyway, the short conclusion is both number of particles are lost and also surface proteins are damaged. Next, we question whether this, the EVs going through these hypertonic cycles, are they stable in the longer term? So we measure the EVs as a function of days, like up to about 1 month. As you can see in the beginning, the tonicity method is very similar to the initial input sample, while the EVs came out from the solidification or exclusion, they pretty much like reduced. But also the decay, you see that when we normalize per input original sample concentration, the decay is actually more steep for the solidification and exclusion products. So yes, we can confirm that this method is actually pretty gentle to engineer to load some materials to your EVs. Next, we also tested whether this dose loaded the EV, whether they are more functional inside of the cell treatment. Yes, both when we treat the EVs with the same initial volume or same number of particles, both cases we see that more cytotoxic efficiency of our tonicity controlled engineered EVs. This was for the dose loaded case, and when we loaded the microRNA, we also see that compared to the control and the RNA-only control case, we see the significant decrease in terms of their cell proliferation efficiency, which means more amount of microRNAs are loaded on the EVs. And the beauty of using EVs is actually bigger and when we use the 3D structure like a spheroid and compared to the not just conventional cell culture. So here we treat the engineered EV on the spheroid. And as you can see, compared to just the DOX case, the EV case shows much more significantly enhanced effect. So we could see the EV is more actively going inside of spheroid, you can imagine. So with that, I'd like to conclude that this ExoBeads, which is an extremely powerful device to have like media exchange without losing the particles too much. And it can be also used as a tonicity control media so that you can load some exogenous materials to the EVs. And it can only work with a small volume of sample to save your precious drugs or some therapy materials. And this tonicity change things, and it will be safe. And we could load many different kinds of molecules like those and different molecular vector testrons and single-strand DNA, etc. So we are currently working on whether we can also use it for guide RNA production, etc. And compared to the conventional method like sonication and extrusion, I think this control method developed by Acilis is a bit gentler. And with that, I'd like to thank you, my group, and the beautiful members from Ulsan, Korea. And I'm happy to answer questions if you may have. Thank you for your attention.
Question? Yes.
Thank you for this nice presentation. I have a question regarding the method you are using to discard or remove the free cargo. Maybe how you separate the non-cultivated cargo from the associated one?
Can I remove the uncapsulated molecules? Yes, that's a very good question. And yes, if you imagine you have filtration, right? So we can retrieve the particles on top of the filter. Those are EVs loaded blocks, the EVs with the loaded EVs. By doing washing, we can remove unloaded materials from underneath of the filter. So we can actually compare those two. In addition, when we loaded RNA, we can also treat the RNAs with a washing buffer. So you can wash your EVs with RNAs together. So you can actually destroy all those free RNAs. And we compare all those, and it's actually very nice to compare these EVs inside and outside as well. Okay. Thank you.
Any more questions? Yes?
Okay, thank you very much for a nice talk. I was interested in the numbers that you showed for loading actually. I see like you load about 10 to the power of minus 21 moles per EV. Can you maybe give like an idea about how many molecules this is?
Yes, we calculate, yes, so that's actually a good question. And again, I must say, first of all, it's all estimation because we only count the molar concentration and calculate the concentration. It's about 10-ish. It's a short answer question. I'm not really sure how accurately I can claim the number. That's why when we use the large molecule, actually the number looks small, but it's actually larger. That was quite interesting when you compare to this conventional inflation.
Thank you.
Thank you for your presentation. I just have a question. So based on the mechanism of the loading, you first expose the EVs with the hypotonic solution and followed by Dox loading. I just want to ask how...
We mix the box inside the hypotonic solution, so it all comes together.
Okay,
not in a sequential.
Okay. So would the addition of doxorubicin, how much doxorubicin is added in the solution? And would the concentration of doxorubicin affect the tonicity or the osmolarity of the solution, which affect your loading efficiency?
Yes, good question, but the concentration is not that high. We confirmed it doesn't change much.
So just one more, have you actually tried to do release testing, release profile testing on doxorubicin, or even characterize the extent of how much EVs contain the doxorubicin?
Yes. But as you can probably know, doxorubicin is a process intensity, so by having calibration curve, we estimated how much is inside and outside, and that's how we calculated the efficiency of loading efficiency. Thank you. Yes.
Thank you.
Okay, I can see on my screen.
Okay.